This presentation is on DNA structure and replication. For this specific presentation, you're going to want your fill-in-the-blank notes, which you can find in the classroom materials bin. Heading into this presentation, it's important to understand that DNA has the same code and structure across all living organisms on the planet, which unites all living organisms on Earth. Remember that DNA stands for deoxyribonucleic acid. It's made up of long chains of nucleotides. Each nucleotide has 3 parts: a phosphate group, a deoxyribose sugar, and a nitrogen-containing base. Notice that T always pairs with A and C always pairs with G. So here you see one nucleotide. You have the phosphate group, the deoxyribose sugar, and the nitrogen combining base. This can change depending on the type of nucleotide base we're working with, and we're going to get to that in a second. What I want you to notice is that this structure never changes shape, no matter what direction the DNA is going in. DNA always goes from 5' to 3'. So we have our 5' to 3'. If you notice, the sugar is always pointing in the direction of the 5' end, no matter what strand we're on. This can help us determine which side of the DNA we're actually on. These are our nitrogenous bases. The nitrogen-containing bases are the only difference in the 4 different types of nucleotides. There are 2 major groups of nitrogen bases. Our pyrimidines, which only have 1 ring, and our purines, which have 2 rings, or a double ring. Our pyrimidines include thymine and cytosine, and our purines include adenine and guanine. Remember that thymine will always double bond to adenine, and cytosine will always triple bond to guanine. Once we realized that DNA was controlling our hereditary characteristics and different events and proteins that were happening within our bodies, we were able to learn a little bit more about what it actually looks like. Watson and Crick determined the 3-dimensional structure of DNA by building molecules. It took them a long time to figure out that DNA is a double helix made up of a sugar-phosphate backbone on the outside with their bases on the inside. They weren't the only ones that worked to help understand the structure of DNA. Watson and Crick's discovery built on the work of Rosalind Franklin and Erwin Chargaff. Keep in mind that Watson and Crick actually thought DNA was a triple helix, not a double helix, thinking it had 3 strands. It was Rosalind Franklin's X-ray images that suggested DNA was a double helix of even width. Unfortunately, she did not get credit for this until later in life. She even passed away before she was honored a Nobel Prize for her work. Erwin Chargaff created rules that stated A will always pair with T and C will always pair with G. One way you can remember this is that the apple falls from the tree and the car goes in the garage. Nucleotides always pair in the same way. The base pairing rules show us how nucleotides always line up in DNA. Adenine always pairs with thymine, and cytosine always pairs with guanine. Because a pyrimidine, which is a single ring, always pairs with a purine, which is a double ring, the helix is going to have a uniform width. The backbone of DNA is connected by covalent bonds, so that's between the sugar and the phosphate groups. These are always going to be covalent bonds, in addition to the bonds between the nitrogenous base and the deoxyribose sugar. The bases between cytosine and guanine and adenine and thymine are hydrogen bonds. That's what's holding them together. So it is easier to break these hydrogen bonds apart than to try and break these covalent bonds. Now that we understand the structure, we're going to talk about how DNA is replicated, because the structure really plays a big role in the function of DNA replication. DNA replication copies the genetic information. A single strand of DNA will serve as a template strand for this new strand that will be made. The rules of base pairing, as defined by Erwin Chargaff, are going to direct the replication because adenine always pairs with thymine and cytosine always pairs with guanine. DNA is replicated during the synthesis or S stage of the cell cycle. Remember, in the cell cycle, we go G1, S, and G2. All three of those are part of interphase. Each body cell gets a complete set of identical DNA. DNA only serves as a template. Many enzymes and other proteins are actually required to do the work of replication. Different enzymes are going to unzip the double helix. Free-floating nucleotides are going to form hydrogen bonds with the template strand. Several DNA polymerase enzymes bond the nucleotides together to form the double helix. The polymerase enzymes form covalent bonds between the nucleotides in the new strands. Notice in this image down here that our original strand is blue. We see that we have unzipped for our replication fork, and the new strand, shown in orange, is being added complementary to the template strand. DNA replication is semi-conservative. That means that two new molecules of DNA are formed, each with an original strand and each with a newly formed strand. DNA replication is fast and very accurate. DNA polymerases can find and correct errors. As it moves along, it may notice an error previously, and it can actually backtrack and redo, almost as if you are editing your own document on Google Word, and you can go back, edit it, and now go forward again and fix it. As I said, DNA is very fast. If you look at the 4 points below, A, B, C, and D, it's showing a sequence of events. A is showing you 3 different origins of replication in eukaryotic chromosomes. B is showing how they start unzipping DNA. C shows the progressive DNA replication. And D shows the newly formed DNA strands, each with an original copy and each with a new copy. Because there are many origins of replication in eukaryotic organisms, DNA replication happens much faster than if there was only 1 point in the DNA on each chromosome for DNA polymerase to do its job. Right, that's the end of our presentation. Make sure you have all of your notes and complete the next assignments on Schoology.